Well, hello everybody. In this video lecture, we will cover physiology of muscle contraction. And it will be two videos because I don't want to make them very long, right? So that's the first video on physiology of muscle contraction. And to do it, I'm gonna use this PowerPoint, right? Okay, so let's go ahead and begin. Well, first we want to talk about three types of muscle tissues. We have skeletal muscles, we have smooth muscles, and cardiac muscles. So let me pick up the pointer. Okay, so here the skeletal muscles or striated muscles, smooth muscles that do not have striation, cardiac muscles, they're also striated. Cardiac muscles only found in the heart. So the structure of the skeletal muscles are long, cylindrical, multinucleated, striated, and they are voluntary. Voluntary means that you control their action, located in your body, right, attached to your bones or to your skin, if those are muscle-facial expression. Those muscles contract quickly, but can't remain contracted for long, so they get fatigued or tired pretty quick. Smooth muscles, they are spindle-shaped, they're uninucleated, no striation, located in the walls of hollow organs, such as your blood vessels, urinary bladder, uterus, stomach, all GI tract, and they contract slowly but remain contracted for a long time, so they don't get fatigued easily, and they are involuntary. So you cannot control their action, their movement. Cardiac muscles, they are branched cells, they uninucleated, the same as smooth muscles. They do have striations, the same as skeletal muscles, and they show rhythmic contraction and relaxation, only found in the walls of the heart, contract quickly and rhythmically, and don't get fatigued or tired easily. They also involuntary muscles. So here's a special characteristic of muscle tissue: excitability, responsiveness, or irritability. So excitability is ability to receive and respond to stimuli. Contractility, well, for skeletal muscle and for smooth muscle, yeah, we use this definition. Contractility means ability to shorten when stimulated. It's not the best definition because we will cover in this chapter that muscles can contract and actually get longer, not shorter, right? But that's how this definition is used for our muscles. Another characteristic, extensibility, ability to stretch, and elasticity, ability to recoil to the resting length. So excitability, contractility, extensibility, and elasticity are special characteristics of muscle tissue. Muscle functions, movements of bones, movements of your maybe your skin, if we're talking about you smiling or frowning. It's also movement of fluids in your body, for example, blood. So any type of movement, actually, right? Oh, well, I would say gross movement of your blood, movement of the lymph, movement of chyme in your GI tract, movement of bolus from your oral cavity to your stomach through the esophagus, right? All these type of movements plus your external movement. So you can walk, you can, jump, you keep your posture because of your muscles. So here, by the way, maintaining posture and body position. Muscles also stabilize your joints and they generate heat, especially skeletal muscles. Contraction. So what is contraction? Contraction is generation of force. And when muscle contract, they generate force that we call, okay, so muscle contract and generate force called tension. Yes, sorry for that. So muscles contract and they generate tension. And tension is pulling something, right? Your muscles actually can only pull, they cannot push, right? So the only force that your muscles generate is pulling force. And you see over here, contraction does not necessarily cause shortening of the fibers. Sometimes fibers are actually get longer during contraction, and we will discuss it a little bit later. But shortening occurs when tension generated by cross bridges between our actin and myosin exceeds the force opposing shortening. So over here in this example, you can see the muscle, right? There is some load. This one is 3 kilograms at about more than 6 pounds, more than 6 pounds. Yeah. So when muscle cells inside the belly of this muscle, when they contract, right, they generate force that pull this load, right, toward the origin of the muscle, right? So now it depends how strong this opposing force is. We can either move this load, or sometimes this load is too heavy that muscles contract, right, but load is not moved. In this situation, muscles contract, they generate tension, but they do not, the muscle stays the same length, it does not shorten in this example, right? And over here, muscle shortens. So contraction is just generation of force. It's not necessarily shortening of the muscles. Sliding filament model of contraction. Okay, so over here you can see the diagram, micro photograph of skeletal muscles, and we see these dark lines, right, light lines, and these darker, thinner lines called Z-discs. So this area between two Z-discs is the sarcomere. Sarcomere is made of several proteins. Some of them are this one shown in blue, that's actin, a thin filament, and this one is myosin or thick filament. And myosin is attached to M line, and actin is attached to Z disc. And you can see how myosin and actin overlap over here. And to contract the muscles, what we need to do, we need to make the sarcomere shorter. If each sarcomere shortens, the whole muscle shortens, right? So this picture number 2 below, you can see that sarcomere now is short. And this has happened because myosin kind of pulls actin along its length towards the midline, right? So in the relaxed state, thin and thick filament overlap only slightly, right? So here's our overlapping area. During contraction, myosin heads bind to actin, so those heads bind to actin to propel the thin filaments towards the M line. So it's it's pulling or pushing this thin filament towards M line. And then H zone, so here's the H zone, and H zone includes only this thick filament. H zone is actually getting shorter. Over here, we don't even have it because we have complete overlapping of actin and myosin. And remember, H zone, we don't have actin. So here, H zone disappears, right? So it became shorter, and sarcomere shortens, muscle cell shortens, and the whole muscle shortens. So make sure before you watch this video, you watch my video on anatomy of skeletal muscles. And in that video, I explain in details all these zones and lines and all these contractile proteins, right? So watch video about anatomy of skeletal muscles before watching video about physiology. Okay, so the 4 phases of muscle contraction include activation. So we need to activate skeletal muscles. Your skeletal muscles cannot contract without stimulus from your nervous system. So that will be activation. The second phase will be excitation, contraction, coupling. So you activate, you excite your muscles, and you start the contraction. That's what happened over here. After activation, muscles are excited and they're preparing for contraction. And this step actually ends when calcium is released from sarcoplasmic reticulum into cytoplasm of muscle cells. Right? We will go over detail in a minute. Next stage is contraction and then relaxation. So on this diagram, you can see that's a motor neuron. And this is axon of a motor neuron, and those called axon terminals, right? So here, 1, 2, 3, those are axon terminals. Axon terminals actually it has this connection, not direct connection, because there is a junction in between with the skeletal muscles. And this area here, right, this area is called neuromuscular junction, right? So skeletal muscles are stimulated by somatic motor neurons, and each axon ending forms a neuromuscular junction with a single muscle fiber, right? So here, our neuromuscular junction. So neuromuscular junctions, they are situated midway along the length of a muscle fiber. Axon terminal and muscle fiber are separated by a gap called synaptic cleft, and this is gel filled space. Okay, so let me kind of show you a picture first, right? And then we get back over here. So here we can see axon terminal. Where's my pointer disappear? Okay, let me get my pointer back. Okay, so that's the axon, right, of a neuron. And this is muscle cell. And this is the junction in between them called neuromuscular junction, right? So there is no direct physical connection between axon and muscle cells. There is a gap, and this gap is called synaptic cleft, right? Now, lots of stuff happening in a neuron that, of course, you will learn when you cover nervous system. But right now, just let's look at this specific event. We will have electrical current moving along the axon. It's like electricity is moving along the membrane of the axon. And when this electrical impulse that we call action potential reaches this axon terminal, something happened. Actually, what happened, calcium moves in, and when calcium moves in, it allows these vesicles to move toward the terminal, fuse with the membrane, and release a special chemical inside this cleft. And this chemical is called neurotransmitter. And this specific neurotransmitter is acetylcholine. So here's our neurotransmitter, right? Then acetylcholine has receptors on the membrane of muscle cells, right? This part is called motor end plate. So acetylcholine attaches to its receptor, right? And when that happens, another channels are open, sodium channels open, and sodium moves inside, right? But so far, this is what we have on this picture. So now let's get back over here. So neuromuscular junction is situated midway along the length of a muscle fiber. That makes sense, right? Axon terminal and muscle fiber are separated by gel-filled space called synaptic cleft, right? So here we have our synaptic cleft. Synaptic vesicles of axon terminal contains neurotransmitter acetylcholine, right? So here's our synaptic vesicles. Here's the synaptic vesicles of the axon terminal contains acetylcholine, that is neurotransmitters. Junctional folds of the sarcolemma contains acetylcholine receptors. Okay, here you see this. This is our junctional folds that form this end plate, and they have receptor for acetylcholine, right? That makes sense? This actually, the process we're discussing right now is activation, right? So that's activation. So nerve impulses arrive at axon terminal. Acetylcholine is released and binds with the receptor on the sarcolemma of muscle cells, and electrical events lead to generation of action potential. Okay, so let's look over here. And again, I lost my pointer. So here's our motor neuron. This is a neuromuscular junction, and this is just magnified, right? That's axon terminal. Here, acetylcholine is green, right? But don't make it confuse you. We can use any color. So acetylcholine attaches to its receptor, and what happened, actually sodium start moving in. And when sodium start moving inside a muscle cell, think about it, sodium has positive charge, right? Now, remember, we discussed it at the beginning of the class, that all your cells, they are negative inside and positive outside. And we can measure this voltage, and it's about minus 70 microvolts. So cell is negative inside, but now when we have sodium moving in, right, what's gonna happen? This membrane inside became more and more positive, right? It moving towards a positive charge, right? And this is called actually depolarization. And so this, when acetylcholine is released and binds to its receptor, we make the membrane inside less negative and more positive, right? And this leads to generation of electrical impulse again. And this electrical impulse is called action potential. So that's what it says. Electrical events lead to generation of action potential. So on this picture, you also see pretty much the same. That's neuron, and this is axon of a motor neuron, and this is axon terminal. And here the acetylcholine is released. So here's the acetylcholine is released. But acetylcholine need to be removed quickly from this cleft, synaptic cleft. And it's done by a special enzyme that called acetylcholine esterase. Acetylcholine esterase, so right here. And what this enzyme does, it's degrade the neurotransmitter. So over here, that's our acetylcholine esterase, and acetylcholine is hydrolyzed into choline and acetate. And this choline is taken back inside the axon terminal, and it can be recycled back, right? And more acetylcholine neurotransmitter is synthesized. So we need to remove acetylcholine quickly to prevent continued muscle contraction in the absence of additional stimulation. So you don't want your muscle to contract constantly, right? You want them to contract only as long as you need to, right? If acetylcholine is present here, your muscles will continue contracting with assumptions that we have other stuff like calcium and ATP. Okay, so I'm gonna skip some of the slides, and I'll go to this picture and I explain you what's happening here. And then you guys do have this PowerPoint on Canvas. You can read it, because whatever I'm gonna tell you right now, right, everything is written in these slides. So, okay, let's go ahead and see what we can figure out based on this diagram. Axon terminal, right? We already talked about it. My pointer, sorry. Okay, so here's axon terminal. This is a vesicle inside with acetylcholine. So acetylcholine is released inside this synaptic cleft. Acetylcholine attaches to its receptors. So here, those are proteins. And here's acetylcholine is attached. And what's happening? Sodium start moving in. Yeah, potassium does moves out, but way more sodium moves in. So we can ignore this potassium for a minute, right? And as I told you, this cell inside is negatively charged, right? But when sodium start moving in, we changing this polarity. Now, over here, we have now positive charge, right? Instead of negative. And when we flip this polarity, when cells became positive inside and negative outside, that's what we call action potential, right? So that's our action potential. So this graph actually shows us very nicely what's going on. So this is the resting cell. So the resting cell has resting voltage minus 70 millivolts, right? This is called a resting membrane potential. Now, when sodium channels open and sodium start moving inside the cell, sodium is positively charged, right? So the voltage became less and less negative. So it's moving towards minus 55. This minus 55 is called threshold. So when the voltage reaches minus 55, another sodium channel open. And those sodium channel are actually voltage gated sodium channel. And the cells depolarize very quickly. So the cells became positive inside, right? And then when it reaches about plus 30 millivolt, sodium channels close and potassium channel open. So now potassium will move out of the cell. Now we're losing positive charges, right? So we became again negative, right? And even kind of overshoot it. This is called hyperpolarization. And then back to a normal state, right? Does it make sense? Okay, let me pick up the pen. Okay, so where I'm gonna draw it. So let's say that's the cell over here, right? So here's the cell. And remember, cell is negative inside and positive outside. Right? When stimulus is received, what's gonna happen? Sodium moves in, right? So when sodium moves in, we have positive charges moving in. So right now, our voltage is minus 70, right? Resting potential. So over here, we have minus 70. When sodium moves in, so positive charges in, what do you think gonna happen with this minus 70? Well, it became maybe minus 69, then maybe minus 65, right? So more and more positive charges moving in, right? So that's what it's shown over here. We go from minus 70 up. And when we're reaching minus 55, the whole bunch of sodium moves in, like lots of sodium, because different channels open, voltage gated channels open. So much sodium moves in, so many positive particles move in that we're moving all the way up to plus 30. Now we have positive charge inside. Well, here's the continuous. Now this channel are closed, right? No sodium in and new channels open. So potassium out. So potassium start moving out. So when potassium moving out, oops, that's plus, right? Now we're losing positive charges. When we're losing positive charges, cell became negative inside again. So that's what this graph shows us, right? And then we kind of restore back to normal, to normal, not this normal, but this normal. Because minus 70, that's our normal resting membrane potential. Yeah? Did it help? I don't know, seems like lots of mess over here. Erase all. Okay, so let me go back here and so everything we discussed so far was our activation. Now we're moving to excitation, contraction, coupling. Right? So let's see what happened next. So excitation, contraction, or EC coupling is a sequence of events by which transmission of action potential along the sarcolemma leads to sliding of the myofilaments. So we don't have sliding yet. When we have sliding, that will be contraction, but we will be ready for this sliding to take place. And latent period is a time between action potential initiation and beginning of contraction. Okay, so let's again get to the diagram. Okay, so let me pick up my pointer. So this is what's happening over here. So we already have excitation, right? We excited this cell. So now this electrical current is moving, electrical current that we call action potential is moving along this membrane of the skeletal muscles, and it moves down the T tubule, right? Now we have this proteins over here in the membrane of T tubules that are sensitive to a voltage change. So when the electricity, well, we don't really call it electricity, we call it action potential or electrical current, right? But it just makes more sense if I call it electricity. I think students understand it better, but whatever, right? So action potential or electrical current moves over here. And what happens with these proteins, they pull on a gate inside the sarcoplasmic reticulum. So inside the sarcoplasmic reticulum, we store calcium. So when we open this gate, calcium is released from this terminal cistern into cytoplasm, right? So here we have calcium inside the cytoplasm. So calcium ions are released. Now calcium ion in the cytoplasm, all this pink, color represent the cytoplasm of a skeletal muscle cell. And what we have over there? We have our actin and a myosin. So myosin has these myosin heads, and myosin heads want nothing more but to be attached to actin and pull the actin along its length. So pull this actin towards the M line, right? But myosin cannot attach itself to the actin. Why? Because there is another molecule over here called tropomyosin that closes these binding sites. So let me pick up the pen. Right, so look, see this structure here and this is the binding site for those heads, for myosin heads. So myosin heads need to be attached to these binding sites, but they cannot do it because we have this brown molecule that blocks this binding site, and this is called tropomyosin. And again, in my first video on anatomy of skeletal muscles, I explain it in more detail. So watch that video before you're watching this one. So now we need calcium, because calcium gonna be attached to another molecule, troponin, and when that happens, tropomyosin can be moved. Look here. So now calcium is attached, so it binds to troponin, shown here in yellow color. When calcium binds to troponin, what happened with tropomyosin? It moved. So now these active sites are exposed and ready to bind to myosin heads. Right, so now myosin attaches itself to the actin, and this is called cross bridges. So cross bridge is formed and contraction begins. So this is our excitation, contraction, coupling. Don't want to keep it. Right, so this is the next second step over here, excitation, contraction, coupling. So activation happens when we receive impulse from neuron, right? And we create this electrical current that we call action potential. When action potential is created, calcium is released. When calcium is released, we're done with excitation, contraction, coupling, and we begin contraction of the muscles. Right, so let's look here. All right, so here's the myosin head, right? So let's see, pointer. So here's the myosin head. It can attach itself or form this cross bridge with actin because calcium is present and tropomyosin is moved away from this binding site, right? So cross bridge formation, then power stroke. So what happened actually, myosin has this ADP and phosphate attached, and it's kind of losing this, right? They are removed, ADP and phosphate, and this causes myosin to push the actin in the middle of sarcomere, right? And this is called the power stroke. Now, myosin will stay attached to actin until we have ATP. So ATP need to be attached, and this will release this cross bridge. And then, well, it releases the cross bridge, and then ATP hydrolyzed into ADP and phosphate. And this stage is called caulking. So that's a myosin head is ready to reattach again and pull again. As long as ATP is present and calcium is present, this process repeats. right? So here's kind of like in every stage discussed again, right? So stage number 1, cross bridge formation, cross bridge between myosin head and actin, right? Power stroke, myosin heads pivots and pulls thin filaments toward midline. So here's, look over here, that's a myosin, right? We have already cross bridge formation, and now we pull this actin towards M line. Because the whole reason why we're doing all this stuff, we want to make this sarcomere shorter. And making it shorter, we're making muscles shorter, right? Muscles contract. So that's our power stroke. Then cross bridge detachment. So ATP attaches to myosin head and cross bridge detaches. And then corking of the myosin head. And corking means this head in a high energy form. It's ready to form the cross bridge, right? Again, so the process can repeat. So energy from hydrolysis of ATP coax the myosin head into high energy state. Okay, and as long as we have everything we need, calcium and ATP, we continue this process, right? That's contraction of the muscles. I also have very short animation here. So let's see if we can look at this animation. I'm gonna mute it because for some reason it doesn't work. The sound doesn't work, but this is 4 minutes. I hope I have enough time. So let's look, and I will explain what's going on over here. So what we have, this is a skeletal muscle. Inside we have myofibrils. So here's our neuromuscular junction, right? So that's our neuron that brings the stimulus to the muscle cells, right? Okay. So inside the muscle cell, we have myofibrils. And inside those myofibrils, we have repeating units, sarcomeres, right? From one Z line till another Z line, right? So that's our sarcomere. And sarcomere made from thick filament and thin filament, myosin and actin. And look, when they slide along each other, sarcomere shortens, right? So here we have myosin, right? And here's the M line. And what myosin, so here's the actin attached to Z line or Z disc, right? And here's the shortening of the sarcomere on both sides. So you can see here how myosin head are attached to actin and how they slide actin towards the midline, forming these cross bridges. Right? That's a power stroke. So right now, cross bridge, power stroke. Corking, cross bridge, power stroke, corking. Right, so that we need ATP to do it. Hydrolyze the ATP and the head in a, ready to attach itself to the binding sites on actin, right? Power stroke. Okay, so let's see. And again, I'm sorry, but I will pause this video for you to watch, but I cannot use the sound. So ATP attach, so we disattach the head, right? So ATP, disattach, right? And attach again. But we need calcium to do it, right? So here's the calcium. And here's our troponin, and this is tropomyosin. You see how tropomyosin is blocking the binding site? But when calcium is attached, right, so here's takes time for calcium to attach, and then tropomyosin is moved. So now myosin can bind to actin and pull it towards the midline. Okay, so as I said, I'm gonna pause this video also, guys, for you to watch on Canvas. And that's it for this video lecture, and I hope it was helpful.